Australia to set up a register so that we really know what the history of a spinal cord injury is. And as you know, not just this movement and sensation. I just wanted to make that point, that that's another aspect that is going to be essential when we think about treatments and cellular treatments. We need to know exactly what the issues are. Next slide, please. So the other issue, you gather then that there are biological issues about when to put cells in. There's the cellular issue about what kind of cell you're putting in and might be the most appropriate. And then there's what's happening in the spinal cord issue. And then there's what's the normal recovery happening and how could the cell change that recovery. And I plotted out here on this scale. You can see it's not quite a linear scale. It goes from 1 day out to 8 years, with those bars showing you roughly when people when these clinical trials that on that clinicaltrials.gov, when they're putting the cells in. And you can see they vary greatly. So these trials here, everybody after here, those are essentially in, particularly if they've chosen Asia A, those will be trials in which the chronic, stable, complete injuries with no further recovery, normal recovery, whereas these ones will be being put in during a period where there's a lot of potential normal recovery. This becomes an issue for patients, but it also becomes an issue with thinking about clinical trials. Again, we can't just say, well, just whack it in, and I don't care what kind of a stem cell it is. There are real issues that, because this is really, it's usually not just a decision between somebody with a spinal cord injury and their doctor. This is highly regulated in Australia, so that to run a clinical trial, you need to you need the patients involved, you need the clinical trials team to be involved, you need ethics approval from the hospital or the universities, you need approval from the Therapeutic Goods Administration who are approving any kind of new treatment. And these are the issues that they have to confront. You've got acute injury on the left and chronic injury on the right. And at the acute stage, you might say the spinal cord is very plastic. There's a lot of things happening there. And hence, it might be a good time to put some cells in there, because if things are happening, we can prod them a bit and make them happen better. Nevertheless, there's this normal recovery that I talked about. So over time, the prospects for that normal recovery will reach a maximum. So based on maybe that plasticity and things happening, the scar tissue hasn't formed. We don't know this, but you might say, well, maybe it's better to put them in early. We don't know this scientifically, but maybe it might be. Meanwhile, combined with that is that if you're putting things in when the normal recovery is taking place, then it might be an increase. This is a bit deceptive. The risk gets to reach it's low. Acceptable risk might be low here because you're intervening with normal recovery. So that as normal recovery goes forth, you might say, well, now you can put something in there. So ideally, at this end, you'd go for therapies that were non-invasive, therapies that didn't involve opening up the spinal cord and doing some extra surgery there. You might think of stem cell therapies in which they are injected into the blood supply or through a lumbar puncture. That might be more acceptable early on, whereas for chronic injuries, the surgical risk of a spinal cord, opening up the spinal cord and injecting cells, that might be more acceptable. So we have to balance out the risk. the availability of patients, there are a lot more chronic patients, of course, than there are patients who've just had their injuries. But nevertheless, because of the changes that are happening, the normal recovery that's happening, if you intervene early, you need a hell of a lot more patience in order to know whether you've had an effect. Unless, obviously, somebody had some incredible change, but most of the changes that we're going to see, at least initially, are probably going to be small changes. Small changes in movement, small changes in sensation. One would hope you might be able to get some changes in autonomic function to reduce autonomic risk of dysreflexia, or you might get small changes in bowel or bladder control. So smaller changes mean more people we have to test to know that you've really had an effect. So these are all things that need to be balanced when you're trying to design a clinical trial. Next slide, please. So I thought I'd just tell you about our clinical trial. It's now finished. We actually started in our first patients in 2002, and we published the work finally, the second paper in 2008. So that just illustrates, I wanted to illustrate that even these small steps take a long time. And this was done through a university, so the funding is not as, I think Geron, the next trial I'll be talking about, they spent about 40, 50 million, 60 million, I heard Stephanie mention earlier, over 15 years for their trial. We spent about $500,000, $600,000 in cash, plus a lot of time. It was really like a cottage industry in comparison, and that really slows down the ability to speed things up. Next slide, please. So just to tell you a little bit about these cells, they're not stem cells, but they are the supporting cells of the nervous system, one of the kinds of supporting cells of the nervous system. In this case, they wrap around the nerve fibres that go from the nose to the brain and tell us, those nerves are telling us what we smell, and that tissue has stem cells in it, and it's continually regenerating. So these helper cells are helping that regeneration process, and that was the logic. Maybe we could put them into the spinal cord, and they will help the regeneration process in the spinal cord. And these are probably the most well-researched cells for spinal cord injury than any other cell type that's out there, including embryonic stem cells or neural stem cells or others. There's just a huge amount of data there showing that in animals, showing that they are a useful cell and can promote spinal cord injury repair in rats. So that's the other thing. All of our evidence comes from animals, mostly rats, some from mice, and the thing about clinical trials is they're experiments to see if humans are like rats. That was a joke. So here's a scan through the nose. We get these cells by going in through the nostril. An ENT surgeon does this and takes them out from up here. And you can't really see the next picture, but that's a marker. The green is there showing we've taken a piece of this tissue, we've chopped it up. We can identify those cells wrapping around nerves in green. And then the other picture